Henrietta Lacks was an African-American woman born in Clover, Virginia, who with her husband David Lacks moved to Turner Station, Maryland. Soon after her 29th birthday, the mother of 5 was diagnosed with an unusually bad case of cervical cancer and treated in the segregated public ward at Johns Hopkins Hospital. She died months later at the age of 30, but cells taken from a tissue sample on the day of her diagnosis lived on in the lab. These cells, cultured by Dr. George Gay, divided consistently and at an alarming rate, and were soon to become the preferred test subject in biolabs for the next 50 years. These HeLa cells have been essential in everything from the polio vaccine, to space travel, to cosmetics, to the unraveling of the human genome, our current subject. The 1960s marked a new age of cellular study. Scientists were hopeful that they could find a cancer virus and create a vaccine as had been done with polio. After HeLa, it seemed that scientists could now culture other specialized cell lines in the lab for research, giving hope that a cure for cancer was soon to come. Then this guy dropped a bomb on the progress scientists believed they had made. In 1966, geneticist Stanley Gartler presented to the scientific community that HeLa had contaminated nearly all lab samples worldwide.
So Nelson Rees examined the chromosomes of the Russian cells. He found they were all identical to HeLa. It meant only one thing. Somehow, some of Henrietta Lacks' cells had got through the Iron Curtain. And the virus the Russians found turned out to be no more than a harmless monkey virus. She had picked it up on her journey to Moscow.
Dr. Gartler's realization came from the presence of the regulatory enzyme glucose-6-phosphate dehydrogenase in all of his lab samples. This enzyme is associated with populations of African descent. HeLa was the only cell culture in his lab taken from an African-American patient. After these findings were presented at the second Decennial Review Conference on Cell Tissue and Organ Culture, labs worldwide discovered that most of their lab samples were contaminated by HeLa. The bomb made scientists realize that there was a problem with how cells could be identified in the lab. It created a need to genetically identify HeLa so that it could be controlled and that this could not happen again. In June of 1973, a group of researchers at the first International Workshop on Human Genome Mapping were trying to figure out how to stop HeLa contamination through better forms of identification than the G6PD enzyme. They realized if they could identify Henrietta Lacks' specific genetic markers, they could figure out what was and was not HeLa. At this point in time, DNA sequencing methods were new and the field was about to take off. One of the most prevalent figures in the development of DNA sequencing was Frederick Sanger, the father of the Sanger method. The Sanger method is a specific way of looking at how DNA is sequenced, aka what forms the genes. In this method, a new way of separating the individual strains and combinations of DNA was used to split the DNA into its individual parts. From there, it was injected with a dye that could be looked at under X-ray. With the Sanger method, DNA could be looked at more closely, allowing scientists to identify specific base pairs that had mutated. The Sanger method allowed for geneticists to uncover, one by one, the specific mutations behind genetic diseases such as Huntington's disease. In 1985, geneticist Alec Jeffreys developed DNA profiling, which made linking DNA to specific people easy and also rapidly increased the rate at which genetic code could be interpreted and electronically stored. The stage was finally set for the Human Genome Project. The Human Genome Project was a 13-year-long, publicly funded project started in 1990 with the objective of determining the DNA sequence of the entire human genome within 15 years.
This had to be the most exciting, momentous kind of science that humankind could have contemplated up until this point. Reading our own instruction book, it is momentous in the sense that you only do it once. If you do it right, that is. Once it's done, and it will basically allow you to cross a bridge that you will never go back across in the future, from ignorance into knowledge.
Today, it still remains the largest collective biological project ever completed. Funding came from the U.S. government through the National Institutes of Health, as well as numerous other groups from around the world. An initial draft of the project was available in June 2000, and by February of 2001, a working draft had been completed and published. The complete map of the human genome was finished on April 14, 2003. This scientific feat came with ethical concerns. A paper published in 1995, titled The Human Genome Project, Ethical and Social Implications, was one of the first to express concerns about future uses of HGP data. Disease testing and carrier screening to them demonstrated a potential for exploitation by employers and insurance companies. Today, gene mapping is used for many different types of consumer product. Genetic testing is used to find personal ancestry, assess risks for developing and passing on certain genetic diseases, analyzing athletic performance, and more. One of the largest proprietors of this type of consumer product is 23andMe. The company is very controversial. Recently, there is concern over Google parent company Alphabet acquiring the startup. When Alphabet chose to purchase 23andMe, they wanted to make the gene testing service as profitable as possible. To do this, Alphabet started selling the genetic information that people sent into 23andMe. The data is primarily sold to the insurance industry.
But at what cost? There are concerns that these companies could be misusing your private DNA data. Suspicions really kicked off when Senator Chuck Schumer condemned DNA testing companies at a news conference, stating, "Here's what many consumers don't realize, that their sensitive information can end up in the hands of unknown third-party companies. There are no prohibitions, and many companies say that they can still sell your information to other companies."
The thing I think that's important to realize is that there is only a single law that covers the sale or our use of our genetic data today, and that's GINA, the Genetic Non-Discrimination Act. And that covers the use of data by employers and insurance companies. It's definitely an area I would be concerned about because as of today, there's no restrictions on that type of use.
When the data is sold to the insurance companies, they are able to scan for possible health risks that their clients are genetically predisposed to. As Murray and Livney explained in their previously cited 1995 research paper, quote, "If the insurer knows that a client is likely to need certain coverage, then the company either will not sell coverage or will overcharge heavily." With the commercialization of people's genetic information, we ask the question, who really owns genetic material? A question that should have been asked in the case of Henrietta Lacks and the HeLa cell. In the history of genetics, the two main ethical issues have been dealing with the very same question of who owns genetic material and how can that material be tampered with. The same ethical questions in the background of creating Henrietta Lacks' immortal cell line should still be considered today when dealing with the ethics of genetics.